morning to all, to our remote audience and to all our speakers. I hope you already had a nice coffee so we can start with full energies our session on genomic medicine, where we will discuss the research, the medicine, as well as the ethical and societal challenges that genomic medicine is posing. So despite the limitation of this online format, I still hope we can have an interactive session. So I would like to encourage our participants to share your questions with our speakers through the chat. So before we go into the heart of the session, I would like to briefly introduce you the 4 speakers. They are experts in different aspects of genomic medicine. Roderick Guigaud is a coordinator of the Bioinformatics and Genomics program at the Center for Genomic Regulation in Barcelona, and he's a leader of the group on computational biology of RNA processing. And he has been involved in several worldwide genomics projects that we'll also present in his talk. Then I'd like to introduce Tim Hubert, Head of Department of Medical and Molecular Genetics at King's College London, Associate Director, Health Data Research UK in London site, and of relevance for these presentations is also the Head of Genome Analysis of Genome England, one of the genomic medicine initiative in UK that we will be discussing. I'd like to introduce you also to Gulsing Gumus. Her background is in research on fetal and prenatal medicine. And since 2018, she's a research and policy manager at EURORDIS. EURORDIS, for those who don't know, is a non-governmental patient-driven alliance representing almost 1,000 rare disease patient organizations in over 70 countries. And is really representing the voice of millions of people affected by rare diseases throughout Europe. And last but not least, Gabrielle Berthier. She has an interdisciplinary background in international relations, ethics, and human genetics. Her PhD thesis focused on next-generation sequencing policies in pediatric cancer and rare diseases. And lately, she worked as a head of partnerships operations at the company Variant Bio in the US. And finally, I'd like to introduce Marta Solis and myself, both working at the Center for Genomic Regulation in Barcelona, and we will moderate the discussion. So the session will first have a brief presentation from each of our speakers, and then we will have enough time for discussion. But since we cannot see you directly, no, we can talk to you, we will still be interested to know a little bit about you, about your research interest, where you are coming from. So in your screen, you should see the Slido, and I like you to answer a few questions. So through your mobile or your computer, you can connect to slido.com and enter the code that you see here in the screen, 23275, and answer the first question. What country are you joining us from this morning? I hope there is someone there. This is an experiment. If it's not working, then we will go through the first speaker. Okay. Then I would say that we go for the first talk. Oh no, we have an answer, one answer. I hope it's not from Tim, from the UK. Okay, I would, well, Sweden, the Netherlands, UK. 3, but international, Italy, also from my home country. More Italian and Swedish. That's now we don't feel alone anymore. Okay, then maybe I would like to go for these people who are following us. Where do you work? We will also be interested to know a little bit the sector where you work. It's academia, industry, NGOs, funding agency, or other organizations. Other, okay. Fund, we have a funder. We have people also from academia. More funders. And the last questions, also to what is your level of knowledge on genomics? It's advanced, basic, or you are eager to learn? Advanced, so we have some challenges here for our speakers. Basic and eager. Okay, so I would say then we really go to the talks. So we'll first have a talk from Roderick Guigaud, then from Tim Hubert, Gulsin Gumus, and Gabriel Berthier. So I suggest that we go one talk after the other, and then we will have a joint discussion all together. Thank you, Roderick. I give you the word.
Good morning to all, whether it's 5 or more people, and I actually like to thank Kayla and the organizers for inviting me to be in this session. I will actually talk a little bit about the projects in genomics that arose after the completion of the Human Genome Project. As you know, the Human Genome Project was a project that took place mostly during the 90s and was completed, estimated to be completed by the publication of the first drafts of the human genome almost 20 years ago. This was a project that triggered a lot of excitement. There was a lot of expectations and a lot of promises about this project. So this is actually an editorial from the New York Times just a couple of years before the project was completed. But the project was already in full motion, and you can read something like, you will look familiar. So healthcare will shift from a focus on detection and treatment to a process of prediction and prevention. You can imagine having an infant tested at birth and the results that say you are susceptible to disease A, B, and C. So this is what we call really precision medicine, and this was what people were expecting when we were at the end of the finalizing the Human Genome Project, and this was probably the scientific project that got larger attention, larger publicity, larger political support. This was the moment in which the project, the finally completion of the project was announced to the world, was simultaneously by Bill Clinton and Tony Blair. At the time, using some sort of primitive Zoom, right? This was a teleconferencing to announce the project. So because this was expected actually to change medicine and to have a radical alteration in the way we live. According to some, by now, all of us should live for these 150 years. It is clear that some of these promises were not fulfilled. Actually, this is news in the same journal, the New York Times, a few years later. And actually, the title is totally contradictory to what the editorial was anticipated in 1999. Genes show limited value in predicting disease. And why is this? Why was this? Is this because we were wrong and the rationale behind sequencing the human genome is because we believe that the genome encodes the instructions that determine the biological traits of the organism, in particular the susceptibility to suffer particular diseases and others, right? And this is true. We were not wrong about this, but the problem is that these instructions were coded in a way that was much more complex than we had anticipated first. We did not know, and we still do not know, where in the genome these instructions are encoded. And actually, we even know less how these instructions in the sequence of the DNA are translated into the biological traits of the organisms. On the other hand, the sequence that was published, the draft genome sequence that was published 20 years ago, was just the sequence of an individual. It's not even an individual, right? But whatever, it was just a single sequence, the sequence of one person, let's say. And you know that actually what really is important is that the differences between the genetic variation that correlates to phenotypic differences between individuals. And at that point, if you remember, the cost of sequencing first human genome was $3 billion. So it was actually impossible to even consider sequencing the genome of the second individual. So in the post-genome era, right, there were essentially, I would say that within genomics, there has been two main areas of post-genomic projects. One, trying to answer the first question, right, where the instructions in the genome are really encoded. And these are called the functional genomics projects. And maybe the most popular one is the ENCODE project, which started right after the Human Genome Project in 2003, and has as a goal to identify exactly where in the genome are the regions that confer functionality. And there are many other projects like that. So I just listed here a few ones, like Phantom in Japan, the Blueprint in Europe, etc., etc. And ENCODE is actually one of the largest running functional genomics projects. It's in the fourth phase, and the results of the third phase were published like a couple of weeks ago. So the project will be running at least for 20 years, twice as many years as the Human Genome Project. And on the other hand, right, there was this idea to try to understand where the information is encoded into the genome. On the other hand, the other set of post-genomic projects had to do with characterizing the variation in the human genome. So what are the genetic differences that underlie phenotypic differences? And the first such project was the 1000 genome project. So sequencing of 1000 individuals from many diverse ethnic and geographical backgrounds, right? So this genome variation project is one, the capacity of sequencing the genome of many individuals, which was possible because advances in the technologies of sequencing that decreased the cost of sequencing the genome 1 million-fold. Have actually probably the basis of the personal genomics project, which actually has as an aim to link, right, the genomic sequence of individuals with a number of phenotypic traits, mostly related to medical traits, right, mostly related to disease or to response to disease, to response to treatment, etc., etc. And I just listed here a few of them. I think that Tim will go more into detail of this, so I will not go into the details. But there are many projects in many different countries with many different angles, which actually aim to sequence the genome of a large part of the population. And there is one in Europe, I think it's called the 1 million Europe genomes, which aims to sequence 1 million the genomes of 1 million individuals across Europe. But of course, so we had these two main areas, right, of post-genomic projects, right, trying to characterize the function of the genome and trying to characterize the variation of the genome across individuals. But of course, I think what really we want to know is how the variation between the genome of individuals translates into the variation of the function of the genome into these individuals that sometimes translates into the variation of the phenotypes. And so more recently, a number of projects have been started that try to look both, to some extent, at the variation across individuals and how this variation impacts functional genomics. And maybe the most popular of these projects is the GeCIP project. The the Genomics project is a project in which tissues from 1,000 postmortem donors have been extracted, and RNA has been extracted from this sequence, from these tissues, and sequenced, and actually the genome of these individuals has been sequenced. So for the first time, we were able to relate in a large cohort of individuals how the variation in the genome of the individuals correlates to the variation on the expression of the genes across different tissues. And as you know, the expression of the genes in a tissue or in a cell is the first manifestation of the instructions encoded into the genome, right? The way in which these instructions are translated, are interpreted into the cell is by DNA triggering the transcription of regions of the genome, the genes, into RNA, right? So this project is actually very important in the sense that for the first time we were able to do in a multi-tissue way of how variation in the genome translates into variation into the RNA expression of the genes. So the RNA is only one of the phenotypes, one of the molecular phenotypes. We know that the transcription of the DNA to RNA is mediated by a number of epigenetic layers, and there is a project now which is actually the convergence of encode and GTEx, which is called NTEX, which actually aims to monitor not only the RNA in the tissues of the individuals, but also many other epigenetic assays like GeCIP for histone modifications, Hi-C, methylation, etc., etc., etc. One thing that's actually important, because we're talking only about these molecular phenotypes, the first readouts of the genomic sequence, is that, of course, we're interested not on the RNA-seq per se, we're interested in how these changes in the expression change the phenotype of individuals. And something which actually is very important in the GeneText project is that also there are histological images and pathology reports of all the samples from the individuals that have been collected for sequencing. So we're talking about more than 20,000. And for instance, this is an image of one of the tissue samples that has been RNA sequenced, which actually is from a tibial artery, which shows heavy arteriosclerosis. Also on the slides, I think a very important project which also aims to really collect phenotypic information on large cohorts of individuals at scale, which is even much larger, is the UK BioBank. And we have heard these days because the UK BioBank, for instance, data on the UK BioBank is being used to many different recent research projects that have led to quite interesting findings, actually also as well in COVID-19. And actually the UK BioBank collects data about 500,000 UK citizens, and which will be followed by for a number of years. And in addition to a number of phenotypic variants of many very different nature, from basic anthropometric variables like weight, height, etc., to medical history, and actually to biological imaging or histological or essentially imaging of organs like brain, heart, bones, etc., etc. And of course, also the possibility, which is already occurring, of doing DNA and genome sequencing of the participants, and maybe even RNA-seq of some of the samples, probably blood, which is the most. So let me finish. So this is the complex pathway that leads from the sequence of the DNA to the phenotype of the organism. We know that in the DNA, the traits of the phenotype of the organism are really encoded, but the pathway that links this sequence of the DNA with the phenotype is very complex, right? DNA is translated, as I say, transcribed to RNA, which then are translated into proteins, and the RNA for the cells, which organize in tissues and organs, and then the tissues and organs are in systems, and the systems actually form the individual. We really know very little about this pathway, right? How to connect the different steps of this pathway. And I've talked about a number of projects that are going to that direction, in the direction of, for instance, characterizing the molecular phenotypes, the RNA and the proteins, the code, text, and text, all the functional projects. Then other projects that actually aim to collect organ data at different levels, mostly imaging on the individuals. So there is an intermediate step, right, between this molecular level and the organ level, which seems to be missed. And this is actually probably the aim. This niche is probably likely to be filled by this other large-scale project that started a few years ago, the Human Cell Atlas. The Human Cell Atlas has as an aim to actually to characterize the almost 3 billion, I think 30 billion actually, I forgot now, cells that are in the human body, right? I think it's just the aim is a little to raise 1 billion cells, which is a lot, by doing actually RNA sequencing of all the different cells in the human body and to characterize all the different types of cells that exist in the body. This is actually quite important because, as I said, the malfunctioning of an organ may be caused by alterations in the cellular composition of this organ. And until we really don't know which is the cellular composition at the molecular level of all the cells of all organs, it may be very difficult to do therapeutic interventions. And I think that I'm not sure whether I went over my time, but I think that's my last slide. And I would like you to thank you for your attention.
Thanks a lot, Roderick. We now move to Tim Hubert.
Hello. So I'd like to thank the organizers for inviting me. And I'm going to follow on really from what Roderick said. You might think from Roderick's talk that it was kind of hopeless to the complexity of humans and how little we really know, although how much data we've collected, we still understand so little. It's difficult to think about applying this to medicine. But that's what I'm going to talk about, where you can usefully do that. So this is particularly the example in the UK. We've set up something called Genomics England, which is a collaboration with the health service of the UK. Initially was to sequence 100,000 genomes to apply this as genome medicine in the National Health Service. So underpinning that is what Roderick didn't mention, the cost of sequencing. When we first sequenced the human genome, it cost around 3 billion euros for the first genome. And it looked like it was going to be expensive for a very long time, but there was a change in technology which resulted in a massive drop in sequencing costs. So now you can sequence the whole genome for less than 1,000 euros, and you can do it in around 1 day. So it becomes practical to think about applying whole genome sequencing to healthcare. The other point about whether you can actually do anything useful is if you consider the health service and think about, rather than sequencing an individual and predicting their future, which is what many people worry about from an ethical point of view, treat it as a very much more targeted application to help in healthcare. And that's by only limiting it to cases where an individual has an existing medical condition which isn't explained, and limit it to those diseases where it's thought likely that only a few variations in the genetics could be causing those diseases. So we're talking here about rare disease diagnostics, which is frequently caused by a single difference in your DNA, or cancer where it's only a few variants. And the point here is that that in a health service, many times a disease is linked to a particular gene, but there's lots of cases where if you look at that gene in an individual, you don't find any variation. So the variation must be somewhere else. It's once you have gone past testing one gene, the easiest thing to do is then test the whole genome and look for another possible variation that might be causing the disease. This is still very difficult because every individual has about 4 million differences, but there are various strategies for filtering those 4 million differences, most of which have no impact on an individual, and spot the one that might be causing the disease. So that's what we've been doing in the 100,000 Genomes project. We've been trying to take people in the health service who have unmet clinical need in rare disease or cancer, sequencing their genomes. In the case of cancer, sequencing both the genome of the normal patient and their cancer, and in the case of rare disease, sequencing the affected person and their parents if they're available, and comparing those genomes to analyze and produce a diagnosis. So this project's been running since around 2013, and it has scaled up with integration with the health service through NHS England to have sequenced more than 100,000 genomes. And all of those results actually have now, the slide's a bit out of date last year, but all of those genomes reports have been returned to individual clinicians in the health service. So this gives you just an example of a case, just to give you an idea of what the benefit can be. Here's a case of Jessica 4, who has a rare disease condition. And she went through a long series of tests because it wasn't clear what was causing epilepsy. And what we found was a completely different, unexpected variant in a gene that's not normally associated with this condition. And it's actually a transporter of glucose into the brain. So the problem here is not that there's a brain defect, but there's a problem with energy supply. And this suggests a treatment. And in this case, bypass that particular transporter into the brain, change the diet to a low carbohydrate diet. And this has led to significant improvements of Jessica's condition. So here's a case where a whole genome sequence has been able to improve the treatment of an individual. These sort of cases are rare, but in many cases, ending the diagnostic pathway for an individual is a major improvement for them to know exactly what the cause is and what's the likely outcome. So lots of countries have been talking about doing this kind of work, bringing genomics into the health service. It's useful to consider how we've done it in the UK, because we are pretty much unique still. Roderick listed a number of projects. Most of those projects are research projects, or they're just getting started if they're talking about integration with the health service. We're now into 7-8 years, and we have a structure to make this a permanent part of the health system. So there's been very deep integration with public participants, health service staff in terms of engagement, discussion, the ethics, how things should be implemented. There's also been very high-level government support. This began as a prime ministerial project initiative and had support from Number 10 throughout the first few years. Of course, UK has a single payer system, which makes things easier in terms of integration across the whole of the health service. And the whole thing it's not been really a research project. I'll mention research in a minute, but this has been structured as an NHS transformation project to put genome analysis into the health service, with research being an additional benefit rather than a driving force, and with partnership with the agencies. So in terms of structure of ethics, which we'll come to later in discussion, the key thing is that we have a patient panel, we have engagement across all the different aspects of the project, representation on committees that oversee how the project's conducted, and particularly from research, how researchers can access data. Secondly, in terms of agencies, the whole of the health service, all the different agencies, Genomics England is a small new component of the health system, but to make this project work has involved the whole of the health system. But it has meant also we can deploy something nationally. And in fact, it is deployed across all the 4 countries of the UK. It's not just in England. So the future of this is that there's a commitment in the UK to sequence 1 million genomes over the next 5 years. Half of those will be through Genomics England and the health service integration, the future NHS Genome Medicine service. So this is just treating more individuals who have conditions where we think sequencing a genome will help diagnose their condition and improve their clinical care. The other 500,000 is UK BioBank, which has been discussed by Roderick. UK BioBank is not a treatment project. It's very definitely a research volunteer project. And in fact, there is no return of data to individuals in that project. The ethics is completely separate, but those are volunteers, which means that the data sharing arrangements are very different and much more open. So that's the structure. We have an aspiration to do 5 million tests, not just whole genomes, in the health service to really extend the value of this and expand from rare disease and cancer into other types of analysis, such as pharmacogenomics, for identifying the best drugs for individuals. The other side, though, I just want to mention is the research impact of this. So the classic way of doing research, Roderick's talked about the various big projects that have been done at different molecular levels. Biobank is obviously 500,000 individuals. There are lots of research cohorts of volunteers around the world, but frequently they're not very well integration with healthcare system data. And of course, it's not a whole population, it's a set of volunteers, which is not quite the same thing as trying to understand the full spectrum of individuals in a population. So what we've been additionally trying to do with our structure, Genomics England, is to set up the infrastructure so that data can be used directly from a healthcare system. And in some ways, Genomics England and Genomes is just a start of that. We're also, through something called Health Data Research UK, extending this to other health data beyond the few individuals that have genomes. And the idea of that is then potentially, ultimately, we might have access to 65 million records. And that is a different class of data, what's in healthcare records, but it gives a much deeper number of individuals and the possibility to analyze multi-morbidity effects. So just in Genomics England, we have this structure called the Genomics England Clinical Interpretation Partnership, which allows researchers to access this data set, the 100,000 genomes data set, and carry out research. And we have around 3,500 researchers who've signed up for that, around 1,000 actively using the environment over the last year and publishing papers based on analysis. But in terms of the ethics, which we'll now come to, I think the key thing is how we have set this up, because here you haven't got, they're not quite research subjects. People have consented for this data to be used, but they're still people who are being treated in the health system. And so the ethics is based on the idea that the data is not distributed. This is very different to BioBank, where the data can be distributed. Genomics England and the healthcare system, data stays inside the NHS firewall, behind the NHS firewall. And the structure we've set up is shown in this figure. So in blue, you have the data, there's a single data center. Blue shows the sort of cycle for analyzing those data for individual care purposes and producing clinical reports that go back to clinicians. But there's another side shown in green where we've de-identified the data and allowed researchers to log into this environment and carry out analysis. But that's a highly controlled system. You can't take raw data out. You can only take summary level data, and that's controlled through an airlock system, manual review of data. But this has been done before in social science and many other fields where there's highly sensitive data, but not on this scale. Here we're talking about 20 petabytes of data. And so you see HPC there, high performance computing, you have to embed inside this data center, inside this controlled system, the ability to run complicated jobs, the types of analysis you need for genomics. So that's what's been built, and it satisfies what we call the 5 safes. So safe people, evaluating, if researchers are bona fide researchers. Projects get reviewed by the Access Review Committee, which includes those patient representatives. This is described as a safe setting, this environment that you can only log into. Safe output means only data that meets a summary criteria, and therefore does not disclose anything about individuals. It can be allowed out for publication. And safe data. The data has already been de-identified into this system. Now, the high performance computing, when you have thousands of researchers, becomes a problem. So we're now extending this concept, safe computing, the idea that you can do this in the public cloud, but with encryption and the kind of key management that cloud providers now provide makes it so that you can be confident. You can say to patients, the cloud providers really cannot see any of the data. It's still entirely under our control. And then the other thing is that for clinical benefit, the idea that you've run your clinical pipeline, but researchers might find something that the clinical side didn't spot. And in fact, in rare disease diagnosis, only around 40–50% of patients are getting a clear diagnosis because of the limits of our understanding. And so if you can set things up so researchers don't know who anybody is, but if they find some pattern, they can send messages back over the fence to the clinicians to propose an interpretation that there's no existing diagnosis. So this whole structure we're describing as trusted research environment. And in fact, I've been leading a project in Health Data Research UK to define a green paper on what you need to do to set up a trusted research environment. And we're using this in the UK. We're kind of using Genomics England model as a guinea pig for how you might do this across health system data as a whole inside Health Data Research UK and something called the Health Data Research Alliance, which includes all the other healthcare data providers in the UK. So I'll stop there. So the summary is there is enough healthcare value to start implementing limited genome medicine in healthcare systems today. It is possible to do this, and that's what we've demonstrated with the Genome Medicine project, the 100,000 Genomes project. And we've shown through the evidence, the health economic analysis, that there's enough value for the health service, which is why the UK has committed to now make this permanent as the NHS Genome Medicine Service. And we'll start, the idea is we'll gradually build up a feedback loop of research leading to other areas where we can do use genome data in different ways to further enhance healthcare. And secondly, that it is possible to configure data access in a way where you allow both analysis for clinical care, but also analysis for research. That's what we've set up, and that's essential if patients, the public, are going to trust you. There's a lot of distrust about data, and if you can say, well, the data doesn't move, and the only extracts don't identify individuals because they're summaries, people in the surveys we've done are much happier with that. And so this led to this definition of a trusted research environment as a model. And I'll just acknowledge there's, obviously huge numbers of individuals and organizations that have contributed this. It's a big team now. It's about 200 people at Genomics England. From the first year or so, there's probably around 8 or 10 of us. Big organizations like NHS England have been major forces. I haven't talked about the education that you need to do, which has been modeled through Health Education England, Public Health England, where you extend this to viruses, which is very relevant now, of course, with COVID. There've been whole systems set up, genome medicine centers, some of which have now become genome laboratory hubs, and large projects on the clinical side, which have been integrated into this project as well. And then, of course, my home institute, King's College, a lot of this, of course, builds on top of work previously at Wellcome Trust, Sanger, European Bioinformatics Institute, and the other members of this consortium, these various consortium that have made up now Health Data Research UK. I'll stop there. Thanks very much.
Thanks a lot, Tim. We move now to Gulsi Gumons, bringing up the voice of patients. And while we are switching, I would still like to encourage you to send us your questions for the debate all together later.
So hi everybody, good morning. I am Gülçin Gümüş. As Michaela said, I'm a molecular biologist and geneticist by training, and I work at EURORDIS as a patient advocate, and I work there as a research and policy project manager. So my role there is to involve patients in research projects and make sure the patient involvement is appropriate in research projects. So I just want to say that today I'm going to be talking about the role of rare disease patients in driving genomic research projects. I will show you the patient perspective and how to include patients in an effective way. So at EURORDIS, we are, as Michaela said, non-profit patient organization. We work towards improving lives of people that are living with rare diseases. And we present more than 71 countries, including 28 European Union countries. And we have outreach to over 2,500 patient groups. So as you might know, we have more than 6,000 rare diseases, and each one affects fewer than 1 in 2,000 people. It might be a small number, but altogether it represents 30 million people that are living with a rare disease in Europe. So what we do at EURORDIS is to advocate, empower, and engage patients in areas of research, diagnosis, treatment, healthcare, social care, so everything that is going around with the life of rare disease patients. And I will be talking about why is research a priority for people who are living with rare diseases. So because through research, we get a better understanding of rare diseases, we get quicker diagnosis, we get better treatment and better healthcare. And also more patient involvement means more research, which is better targeted to the needs of patients. And then it would mean to develop more therapies that are targeted to the needs of patients. So research represents hope for these 30 million people that are living in Europe with a rare disease and also their families. So what do we do in terms of research at EURORDIS? So before I explain that, I'd like to show you quickly what do rare disease patients think of research. So this is what you see is a rare barometer. It's a EURORDIS initiative survey, and it has more than 3,000 responses. So we launched these surveys in different areas, for example, on care, on data sharing, on access to medicines, and this one is on participation in research. We do this survey to help improve the research that is being done, and also we want to know what is important for patients. So here you can see we ask them what are the obstacles that they think the rare disease research has. So, and what kind of research do patients participate in? And you see that 1/3 of these patients who responded to the survey, they participated in the research, and they're familiar with participating in research. Actually, they have 16% of them joined research to develop genetic therapies. So the top priorities for patients are to develop therapies, to improve diagnosis, and also to identify mechanisms and causes of rare diseases. And this is really important. The last thing is what matters to patients, what the patients want. They are willing to participate in the research project. They're willing to share their data, but they want to receive clear information. They want to receive information on the project that they want it to be explained to them clearly. And also they want to have quality relationship with researchers. So this is essential. They don't want to be considered as guinea pig or research subjects, but they want you to explain them things, giving feedback after the project, showing them project results, and so that they will be feeling that they contributed to research in somehow. So I will show you how do we involve patients in research projects. So at EURORDIS, we have different, we are involved in different projects. I'm just going to show you 2 examples of projects. It'll be easier, otherwise it's just all abbreviations. So I will start with Solvardi. So Solvardi is, well, it's a project with the objective to solve large number of rare diseases for which a molecular cause is not known. So they use combined omics approaches and they improve diagnostic of rare disease patients. So we lead a work package on training and engagement to ensure these patients are engaged in the project. And I will show you that the result of this work package. So this training is called EURORDIS Winter School on Scientific Innovation and Translational Research. So as part of the SolveRD project, we developed this training, and it's basically about how preclinical research translate into real benefits for rare disease patients. So it's a 5-day face-to-face training. We did 3 editions already. So the first edition was in March in Paris. The second edition was also in Paris, and the third edition was online due to COVID. But yeah, we have this 5 face-to-face training, 5-day, and then before that we also have online training for patients to participate, to get to know a little bit more about research. So they will do some pre-training courses, they will do some quizzes and read some articles on research. So when it comes to the day of training, on the day of training, we talk about rare disease research environments, genetics, diagnostics, research, data sharing, and genetic therapies. And why do we do this? We do this because the patient learn about the disease research environment, and they have the opportunities to get engaged in rare disease research. So the more they know why the research is being done, the easier they can link. And so that the patients can understand the scientific process behind the preclinical trials and what is the importance of these preclinical trials. And then the last thing is that so that the patients are empowered and they are valued partners from the beginning until the end of the research project. So this is the SolveRD project that we have, and I will show you the other one, which is the European Joint Program on Rare Diseases, and this is the patient involvement that we do. So it's, I will show you a little bit more later on, but the European Joint Program is the biggest European project that coordinates rare disease research in Europe, and it has over 130 institutions. So they launched this called development of novel therapies in a preclinical setting. So it includes cell therapies, gene advanced therapies, and we are the core member of the governance committee within this program. So we ensure that the patient needs and priorities are represented. So we advocate to make it so that patient organizations can be official partners in research consortia, so they can receive funding through their research calls that are part of this program. So before, they did not receive any calls, and they would be just patients that would be contributing to the projects. But now patient associations can be partners in research project, so that the patient engagement is encouraged. Patient organizations are eligible to become a partner and get a funding to carry out their specific activities. It can be networking activities or other types of activities, or workshops or working groups. And then the other thing that is important is that now patients can act as reviewers in this project. So they will evaluate the patient engagement part of the project. So they will be making sure that the patient engagement is appropriate and it's relevant and meaningful, and then they have the adequate resources allocated within the project. So within this project, we had, we just started a couple of months ago, but then there were challenges for patient involvement. So first we identified the challenges, and then we tried to see how we can solve them. So the challenges that we faced were that, there was a different understanding in patient engagement and patient involvement. So it was quite different. And nobody knew where to start. So researchers didn't know how to approach patients. Patients didn't know how to approach resources. And also there was no baseline for these patients to review these projects. So there was no examples, no guidelines. So what we did is we decided to develop this guideline for patient engagement in research project. And we started this working group. We started this working group for patient engagement in research. So the goal was to draft some guidelines for patient engagement in these research projects, and the impact is to have more systematic and patient-centered research, and to have more improved understanding and the added value of patient engagement in research for the whole rare disease community. So we did set up this working group. We developed our terms of reference. We did teleconferences every 2 months. So our patient representatives got to talk about what should be included in the guideline. And we did a workshop, which was an online workshop. And now we are publishing these workshop consolidating guidelines. So we are going to, in the end, potentially adapt these guidelines as e-learning materials, or also disseminate it for other people to use as a material in their research project, or when they're preparing to apply for a project. So this is the kind of patient engagement we do at EURORDIS. So there are different types of engagement. Well, I just wanted to give you these few examples. So I will just finish with a few take-home messages. So involve patient organizations in every stage of the project. So they are empowered and valued partners from the beginning until the end of the research project. You can involve them in design, in methodology, in implementation, dissemination, and patient involvement, remember that it can be done in various ways, not just for dissemination, but you can invite them in multi-stakeholder meetings, you can create working groups, you can have them review protocols, you can invite them in your activities, speak at your conferences. But yeah, whichever patient involvement you choose, always aim for meaningful patient involvement. And then once you reach for the meaningful patient involvement, you will be surprised on how much impact and quality it will bring to the overall project. So thank you very much.
Thanks a lot, Gulsin. And we have now our last speaker, Gabrielle Berthier.
Hi, everyone, and thank you so much, Michaela, for inviting me to present today. So I'll try to shift to talking specifically about some of the ethical challenges that are associated with genomic medicine. But often when you have these talks about ethical challenges, you kind of get overwhelmed with the idea that genomic medicine is just something extremely complicated that generates so many issues. But what I wanted to do today is not only present those issues, but also talk about a number of strategies that are actually already in place and already explored in order to solve those challenges, or at least tackle them. All right. So just a little bit of an overview of what we'll talk about today. So I'll talk about genome editing and reproductive genetics, a little bit about genetic discrimination and the idea of unequal access to innovation. And then in terms of strategies, I'll explore several from the ethical perspective, legal perspective, societal, or scientific. All right, so let's start with maybe the picture on the right. So this is an article written in 2016 after Holland announced that they would broaden access to NIPT, or non-invasive prenatal testing for Down syndrome. So NIPT is now a technology that is available in many European countries. It is reimbursed, maybe not to 100%, but it is becoming more and more accessible. And what it allows for is early detection of a risk for Down syndrome and other trisomies as well. You know, the reach of this technology is increasing as well to a number of other conditions. So this test can be done very early in pregnancy and through a simple blood test. So it provides numerous advantages, not only for women and families who have access to information at an earlier stage, but also it reduces the need for very invasive procedures such as amniocentesis. So obviously, this also, because it reduces the need for invasive procedures, it has not only advantages for women and families, but also for the health system, because it can provide cost savings. However, this poses a number of ethical questions. So the one that is portrayed in this article specifically is the argument that is made that if NIPT is highly accessible, more and more women will choose to have this simple blood test early in pregnancy. And we may have a situation where more and more women choose not to proceed with the pregnancy in case they are found to have a high risk for Down syndrome or trisomy 21. So this significant decrease in the number of children born with this disease could potentially has ethical implications. So the idea that it would lead to the disappearance of people with Down syndrome down the line, but also a lot of societal consequences, meaning that it could potentially lead to less investment in support structures and understanding of the disease, and it would, in the end, it will have very negative consequences for people who are living with the condition and their families. So this is typically an example where you have a technology using advanced genomic technologies that both has enormous advantages, but also causes maybe ethical concern. And this leads me to the picture on the left, which some of you may recognize this gentleman. So it's Professor He Jiankui from China. So in November 2018, in the picture showed here, he revealed that he had used the CRISPR technology to edit embryos, which were then implanted into several women. And this had led to the birth of twin girls born with an addition of the CCR5 gene that is involved in HIV toxicity. So supposedly he announced that he had allowed for the birth of twin girls with an edited genome, which would allow them to resist infection to HIV. So obviously this generated a lot of outcry in the scientific community and in the broader community. So this event poses a plethora of ethical questions, and I'll name only two of them. First is what is our responsibility as a species to generate human genome modifications in this case, which are transmissible to future generations? So what are the consequences of us being able to redefine in a way how our species evolves? And it also blurs the line between therapy and enhancement. So while one might argue that Down syndrome is a syndrome that is affecting the life of people who are born with it significantly, we can also think about the potential resistance to an infection as something of more of like an enhancement. So this difference between enhancement and therapy is interesting and is really blurred by this new technology and this use of the technology. So I want to point out that I just want to show this very quickly, because there is actually a session later today at 4:15 in the same virtual room 1 on the ethical dimension of genome editing. So if you're interested, I would really encourage you to go there. All right, so now switching over to another issue, which is that of genetic discrimination. So I think most of you will also recognize this little box. So it's a saliva collection kit from 23andMe, which is one of the most well-known direct-to-consumer genetic testing companies. So in recent year, there's been a lot of debate about how the data is collected and used by companies which collect them through direct-to-consumer genetic testing. I want to point out that maybe some of you are aware that recently 23andMe announced that they had to lay off 14% of their staff because of slowdown in sales, and we can argue that this may be caused by an increase in preoccupations regarding privacy and the use of data. So the company has been criticized, and such companies have been criticized for not being transparent about the way that they use the data, for not being transparent about the way people are consenting for their data to be used beyond their own interpretation, but also for further research, and especially research which is done in collaboration with the pharmaceutical industry, which can generate a lot of profit, and this profit is not shared obviously with participants, and sometimes done with participants arguing not to be aware of that being done. So in addition to this question of just understanding how your data is being used by those companies, in recent years we've seen as well that data has been used by law enforcement in the US to identify criminals. So this is the case of the Golden State Killer. So the idea is that through identification of relatives or cousins of a criminal in databases shared by those companies, you can then identify people who had had their DNA collected on a crime scene. So I think that in 2018 there was an article published in Science which projected that at that date, so 2 years ago already, they projected that about 60% of individuals of European descent are actually could generate a match in a third cousin match search within data that is already available on from databases collected of data collected by these companies. So the idea is that the more people get their data collected by these companies, the more data available there is, the higher it is likelihood that you could potentially be identified, even though you never participated individually in this data collection effort. So the general concern, obviously, is that not only if you participate yourself, one might argue that you're not always aware of how your data is going to be used, but also because of this third cousin match identification, you could potentially be identified even though you never participated. Now, access to this genetic information for the wrong reasons can lead to discrimination, which is basically you can be treated unjustly based on your genetic background. So questions on access of this information by insurance companies are raised as well. Now, another issue that I would like to point out today, and it's the last one, after that I'll switch to strategies to tackle those issues, is unequal access to innovation. So on the graph on the left, you can see that although there is progress, still in 2016, only 19% of genomes that had been sequenced were of non-European ancestry. So the vast majority of genomes sequenced to date are of European ancestry. This same idea, these are based on research projects, and this was already in 2016. Now, a more recent article with the graph on the right shows you, displays a number of, each dot represents a biobank that has generated genomic data on these participants. So on the x-axis, you can see the more further to the right, the more recent the endeavor, and then on the y-axis, you can see the percentage of non-European ancestry participants. So you can see that the red dots on the top represent Asian initiatives, so Asian biobanks. You can see that green, so American, and then blue, European initiatives obviously have much of a lesser percentage of non-European participants. I wanted to highlight that authors of this article are associated with the BioMe biobank, which is the dot right in the middle, which is based in New York and has access to a huge diversity in terms of ethnicity. So why is this an issue? Well, because genomic medicine innovations are based on existing data, the drugs and diagnostic therapies and diagnostics which will be developed based on genomic data will be tailored to European populations, and they may not be effective in non-European populations, so this can lead to therapeutic failures, but also misrepresentation of ethnicities can lead to misdiagnosis. So when you analyze the frequency of a variation in a European population, frequencies in non-European population can look very different. So the idea is that nations which have a high proportion of non-European ancestries will have less access to innovations that have been led based on data that is mostly European. Now, in terms of, to go back to the idea of genome editing and strategies that one can take to tackle the issues that it presents, I wanted to show you this picture from an international summit on human genome editing. So the scientific community and governance and policy community has come together and really declared that there should be a lot of caution when using the gene editing technology. So this led to the publication of this article, and I just want to read one sentence from this article, where authors call for a global moratorium on all clinical uses of human germline editing, that is changing heritable DNA to make genetically modified children. So this is exactly what what has been done in China. So you can see that, the global community can come together and call for a specific approach, which then can be adopted by different nations. Now, another strategy that can be used, I call it ethical legal. So this is an example happening in France. The bioethics law in France governs a number of medical procedures, and it also governs the use of genomic data broadly, and it has to be revised periodically. So in the last round of revision, the government organized, a public consultation, very, very large public consultation, to understand what citizens wanted to change about the law and what citizens were willing to discuss. So they organized a total of 271 events to which 21,000 participants, to which there were 21,000 participants. So this is a very broad consultation where they really, brought together citizens to discuss what changes they wanted to bring to the law. In addition to these in-person events, they also had a website which had over 64,000 contributions. There were 183,000 visitors. So it was a very broadly visited website. And I can give you one example of the conclusion that was made related to one of the issues that we mentioned earlier, which is that there was a broad consensus that access to more safer prenatal testing, such as an IPT, was highly acceptable, but that this should also go together with really a reinforcement of policies and also investments and budgets to protect the most vulnerable populations, including those affected by handicap. So really the citizens were in favor of innovation, but within a certain framework, and also while supporting the most vulnerable populations. Another strategy is the GDPR. I won't say too much about it. It's just the idea that you can have a very legal approach to data protection. So it provides this a general data protection regulation, provides really a common European framework for the collection, storage, and sharing of personal data. And most of the time, genomic data is considered personal data, and its storage and sharing is associated with extremely high protections and strict protections. Now, by no means am I saying that this is a perfect solution. So there has been a lot of criticisms of the GDPR for being too strict, too difficult to interpret, leading to a lot of chaos in research initiatives wanting to share data, especially between the US and Europe. But it is an approach that has been taken following the overall recognition of many risks that are associated with privacy linked to the publication of genomic data. Now, another strategy that I would call societal is taken by Dr. Joly at McGill and his collaborators in 19 different countries, which is to establish the International Genetic Discrimination Observatory. So Dr. Joly and his colleagues basically have recognizing that there was a lot of concern over genetic discrimination, but a lack of evidence of genetic discrimination. So a lack of data on how, for example, insurance companies actually do use the data and discriminate based on genetic information, have set up this observatory. So it's, a collaborative research project which will assess policies that exist in different countries and their efficacy, but also the idea is that it provides information for the public. So the public will be able to log in, get information, and also report on cases of genetic discrimination. And this is my final slide. I just wanted to also point out that the scientific community and the research community also has come together to provide very concrete solutions to some of those issues. So I'll mention two initiatives. So the first is the Global Genomic Medicine Collaborative, so the G2MC. This is really has for, the objective is to create a community of leaders really globally, so including low and middle income countries, to enable the implementation of genomic medicine. So they're funding a lot of demonstrating projects. So they have projects, for example, on breast cancer in Rwanda, or on colon cancer in South Africa and the Middle East. So they really provide funding for research on genomic medicine implementation in low and middle income countries. But also the second link that is there, so the genomicspolicy.org, they actually created a complete list of all the genomic medicine initiatives worldwide. So you have information on Brazilian, Mexican, Egyptians, a lot of different initiatives there. So this is really an incredible resource. And the Global Alliance for Genomics and Health provides toolkits, so implementable toolkits to protect privacy, data security, data interoperability, as well as regulatory and ethics toolkits. So they provide guidance on ethics review and consent. So this is my last slide, and I want to thank you for your attention.
Okay, I would like to thank all the speakers for their nice talks. And maybe we have more or less 50 minutes for our debate. So again, I would like to encourage whoever is following us there to ask questions. And maybe to break the ice, I have one question, but also the speakers, if you want to ask us questions to each other, so we try to make this as lively as possible. So maybe I'll start with a bit of provocative questions, and I like to also have the view of everybody. So from the different calls, from your different presentation, it's clear that achieving the benefit of genomics medicine requires sharing data, sharing genomics data, sharing like phenotypic data. And can we think then of a patients actually who benefit from genomic approaches, that he also should have a sort of obligations of sharing his or her data? No, because we know there are lots of debate also on informed consent. And I feel there is a bit of tension between the restriction, the need to regulate, and then to have new legal means, but at the same time, this needs from the genomics to share. And I don't know who wants to start. That would be nice to.
I want to challenge that a bit.
Okay.
So I wouldn't. So we try and avoid using the word share anymore,
Okay.
because share implies distribution, and people get worried about distribution because, as was talked about, that can lead to the data being used for purposes that you didn't intend. And so we talk about access, and what I described as trusted research environments, the basis behind that is the data doesn't move. And that means that you can say much more confidently to patients, your data is being used for research, but it hasn't gone anywhere. And if it has to be combined with, if we want to do analysis across international groups because of number of patients, we can do that by federation. We can federate summary level data. We don't again have to distribute individual level data. And if you say that, I think patients are much happier, public is much happier, and it's the kind of middle ground which partly addresses some of the problems.
And this is what you observe in Genomics England. So people with this narrative are consent to give access to their data for research.
So up to now, the 100,000 was a pilot project which was set up in a slightly different way. It required consent from everybody, joint consent. In future, it will be a separate consent for research. But this has been tested in a big multiple engagement workshops and co-design workshops actually with participants about what, the general conclusion. There's a big one that was published only a few weeks ago from London, a deliberation over 4 days with 100 participants across the whole of London. And the support there was for research, but also support for these kind of controlled environments where data isn't distributed.
Would be nice to hear also the opinions. Gulsin, I think you want to talk.
Yeah. Yes, maybe I can give an example actually from this another survey that we conducted, which was on sharing data while also giving access to data. So we did the survey at EURORDIS with more than 2,000 participants from 66 countries. And we asked them different questions on their perspectives on sharing the data or giving access to data. And these were like patient representatives, rare disease patients and their caregivers, and almost 100 of them were willing to give access to their data to foster research regardless of their age, level of education, or the severity of disease, and also their social demographic profile. And it's really interesting because when you compare it with the general public, this is only like 40% of these people that would like to share their data. The only thing that their rare diseases patients would like to ensure that their data is not being shared with third parties without their consent, that it's not being used within a different consent that they were disclosed, and that it's not used without their knowledge. And that's it. So they're really open and to give access to their data in terms of research.
And Gabriel or Roderick, from your perspective?
Yeah, I mean, from my perspective, I think I really fully agree with Tim. There are technical solutions to this problem, so you can provide an extremely safe and secure environment where the data is stored, and you can provide restricted access to the data where the data doesn't move. But I think also, to go back to what Gultin was saying, the key is transparency. And I think that, of course, providing very transparent and direct information to participants on how their data is being used, is the key to allowing data sharing and making people realize of the incredible value that their data has for other people beyond just themselves. So yeah, just one technical solution and one transparency communication solution. Yeah.
I don't know if Roderick, you want.
I think transparency, as was described, there's a difference between telling people how it's being used and involving them in the decision-making process. And so there's co-design and oversight. And so, our review process, the Access Review Committee is patient participants. So they get to review the use of the data rather than being told how the data is being used.
Roderick, you wanted to say something?
I haven't really giving a thought at this as much as the other participants, but I just want to mention two issues. The first is that because really we do not know how the instructions are encoded into the human genome, the way in which we're actually associating genetic variation to diseases is actually through association studies. This means that we actually have no clue why a gene causes a particular condition. We just know that the gene is associated to this condition. And we know this because we've been able to genotype or sequence large cohorts of individuals which share a particular condition. Now, because most conditions are caused by multiple genes, each of which has very small effect, you really need very, very, very large cohorts if you really want to identify the genetic variants associated to diseases. And essentially, I think we should all be aware that the larger the cohort, the larger the information that's available to the researchers to establish these relationships between genetic variation and conditions and traits and phenotypes, I think the benefit will be larger for everybody. So I think that everybody would agree with this. And there is this project called the Personal Genomics Project, in which there is initiative actually for people to decide to make their own genomic sequence and associated phenotypic sequence available to everybody, which I think it's an option that all these large personal genomics projects should provide to individuals. Now, the second thing that I wanted to discuss, which is a little bit more probably more problematic, is the following thing. So if we are asked for consent to something, this means that you are assumed to be the proprietor, the thing that you are being consented, you are the proprietor of the thing. You own this thing, which means that we are proprietaries of our own genomic sequence. And I don't think that this is something so clear, right? So it cannot be really taken to the extremes, because if there are two identical twins, it's obvious that I am not the owner of my genomic sequence, because if my twin decides to disclose the sequence, it's actually disclosing my sequence automatically. And actually, if my son decides to disclose his genomic sequence, it's disclosing half of my genomic sequence. So this issue of property of the genomic sequence is an issue which I think it's a little bit more complex. And this idea that we own our own genomic sequence, which is behind this idea of the consent. I think it's something that probably would need to be, I don't want to be very provocative, right? But I think it need to, and I said, I haven't given it a lot of thought of this, but I think it's something that we really need to reflect about.
I don't know if our legal ethical expert wants to add a few words, Gabriel, on the property of genomes.
No, it's a great question. I think that it has particular significance when and has been discussed when you talk about specifically isolated group or vulnerable groups. So when you work with communities that are isolated as opposed to communities that are less isolated, the idea is that anyone in the community sharing their data will effectively share information on the entire community. And I think the response to that is engagement and participation. So the idea is that if you co-design research projects using this data, this will be, this will solve the issue of being informed of how this data being used after the fact. Now, I think that the devil's in the details, and the question is, what do you mean by data? Do you mean the information that it provides? Do you mean your identity, your identifiable information? Do you mean the interpretation on your disease risks? I think depending on what information you take from the data, you will have different legal protections associated to those, if that makes sense.
Okay, I think we have time for one more questions. We heard about genomic medicine initiative in UK, in France, in the US. So what about middle low income countries? No, also at the level of research. We also saw the slides from Gabriel. If I saw it correctly, also I didn't see sequencing genomic project on African population or not as high as the other ethnicities. So I would like also to have a little bit your insights, no, or also your reflections, no, on also on genomic medicine in these countries that are not as fortunate and developed and as others.
So actually,
yeah.
G2MC is a good example there because it's got 25 countries at
least. Yeah.
And it's, it spans the world, and there are genome medicine projects in middle income countries. Thailand, for example, and various others in Southeast Asia have genome medicine for particular, things like Stevens-Johnson syndrome. They have a whole screening structure in their health system because the severe effects of not identifying that. And in fact, they've just initiated a genome sequencing project for rare diseases. So the technology is actually cheap enough that it is deployable in a range of different settings at different levels. It doesn't have to be whole genome sequencing.
So are there specific? Yeah, go ahead, Gabriel, sorry.
No, exactly. I mean, I think that the G2MC, so the G2MC produced a catalog that now has 65 initiatives from 35 different locations. If you go to the website, I don't think I need to share my screen, but the genomicspolicy.org has a catalog of information. I think that we should mention H3 Africa. So H3 Africa is a funding mechanism for genomic medicine initiatives specifically in Africa, in the African continent. So it is funded by the Wellcome Trust and the NIH. And it really, so the design of H3 Africa is really to promote local initiatives and local research. So supporting not only medical initiatives, but also local researchers in order to produce information. Brazil also has a genomic medicine initiative, Mexico as well, Chile. So I do think that there are a number of initiatives that are already in place. The big issue is, of course, funding. Access to funding is an issue, and also priority setting. So if you have issues of poverty that are so high that you can't really invest in the health system, then genomic medicine seems like something you wouldn't invest in first. Now, the G2MC provides a number of mechanisms and information on how this can actually help saving costs the health system. But still, you know, I think it's there is still an issue of priority, and also the big issue of, you know, the lack of diversity in the data that is already produced. So if you want to just implement you know, strategies that have been set up in European or Western countries, you can't just, you know, take those directly in sometimes in your own location, because of the lack of relevance of these in non-European populations. So that's a big issue.
Okay, thank you. I think time is running out, so I would like to thank you again for your participations and contributions. And thank you also for the remote participants that we couldn't unfortunately meet. Thank you.